Hello everyone, once again, welcome to the part 2 of the blood glucose regulation. Just before starting, we'll just have a recap on what we have discussed in the part 1. We basically started with the terminology, what do you mean by FBS, PLBS, what is the normal glycemia, hypoglycemia, and all those things. Then we have discussed about the methods of estimation of glucose, where in the standard laboratory procedures, we'll be using GOD, POD method, and the automated machines will be using hexakinase method. And then we have POCT device, that is a glucometer. Then we have discussed about OGTT, how the patient is prepared, what are the indications, contraindications, and all the test procedures. What is the classical method? What is the modern method? What are the differences? How to interpret? What are the abnormal curves for the OGTT and all those things. And then we have discussed about diagnostic criteria for diabetes, who should be screened, who are the high risk population, and all those things. And then finally, we have discussed about glycosuria and glucosuria. What are the differences between those two? And finally, the regulation of blood glucose, a small introduction, where the hormones which increase the blood glucose and which decrease the blood glucose, a small introduction was given. And so this will be the continuation of that class. And so in this class, like I have already discussed, we will be discussing about insulin, incretins, and glucagon. So let's start with insulin. The term insulin originates from the Latin word insula. It means island. It refers to the islets of pancreas. In 1869, Paul Langerhans was the first to distinguish alpha cells and beta cells within the pancreatic islets of Langerhans cells. Then, in 1889, two scientists named von Mering and Minkowski, they have performed pancreatectomy, and because of this they have seen diabetes being produced where their pancreatectomy was done. So experimentally they have proved pancreatectomy can result in diabetes. Then in 1922, working together, Frederick Banting and Charles Best successfully isolated the insulin from the pancreatic tissue. For this, for this discovery in 1923, he was awarded the Nobel Prize. Then the structure of the insulin was sequenced by Sanger in 1954, for which he was awarded Nobel Prize in 1958. So insulin is manufactured and released by the beta cells located in the pancreatic islet cells of Langerhans. Step by step, we will see the maturation. The hormone starts as a large inactive precursor called as pre-pro-insulin. It is made up of 109 amino acids. Remember each of these number of amino acids. From pre-pro-insulin, a 23 amino acid sequence of amino acids called as leader sequence is removed away from the pre-pro-insulin, leaving an 86 amino acid pro-insulin. This proinsulin will be processed in the Golgi apparatus, where the enzymes proteases will be acting on proinsulin, and it will remove the C-peptide, a 33 amino acid uh, polypeptide called as C-peptide is removed, and the remaining 51 amino acid molecule is the active insulin. And the insulin has A chain, which is made up of 21 amino acids, and a B chain made up of 30 amino acids. So pre-pro-insulin 109 amino acids, pro-insulin 86 amino acids, then active insulin 51 amino acids, and C-peptide 33 amino acids. This is in short about the biosynthesis of insulin. Next, coming to the secretion of the insulin. The secretion and the clinical measurement of insulin involves specific molecular structures and predictable concentrations in the blood. First, coming to the molecular storage and the structure. Insulin is stored within specialized granules inside the beta cells. Within these granules, insulin molecules are arranged in the form of hexamers. Hexamers inside the granules. The hexamer structure is stabilized by the presence of zinc ions and calcium ion. And then finally, we will be talking about the daily output of insulin. Daily secretion is approximately 50 units of insulin is secreted by the pancreas every day. Then the fasting blood levels is 5 to 25 milliunits per liter. The proinsulin makes up about 5 to 10% of the total insulin detected in the plasma. That is, these are the 3 points that you have to remember. The daily secretion, which is 50 units per day, fasting blood level of insulin 5 to 25 milliunits per liter, and among the measurement, we find proinsulin in the plasma approximately 5 to 10%. The pro-insulin is significantly less effective than the active insulin, where that is the biological activity of the pro-insulin is one third compared to the active insulin. The and this paragraph includes the clinical importance of C-peptide, the final step where the proteases cleave the pro-insulin into active insulin and C-peptide. What is this C-peptide? Insulin and C-peptide are produced and released into the bloodstream in equimolar quantities in 1:1 ratio. Because of this consistent relationship, measuring C-peptide levels acts as a reliable clinical indicator or index for actual rate of insulin secretion by the pancreas. The half-life of insulin is just 5 minutes in the blood. So if you measure C-peptide, which is having a larger half-life, it is equal to measuring the levels of insulin because the body produces and releases both insulin and the C-peptide in 1:1 ratio. Then finally, coming to the factors increasing the insulin secretion. So the first factors is that as the blood glucose levels increases, the insulin secretion also correspondingly increases. That is the general thing. So how does that happen is that the beta cells, suppose this is the beta cell of the pancreas. The beta cells have GLUT2 receptors. One second. Have GLUT2 receptors. What do they function as? These GLUT2 receptors present on the beta cells, they act as glucose sensors. So they will sense the levels of glucose in the blood. Whenever the glucose level is increased, more and more glucose is taken up into the beta cells, oxidized because an ATP is generated. This ATP will stimulate an ATP binding cassette protein called as ABC protein or also called as ABC protein or ATP binding cassette protein. ATP binding cassette protein, ABC protein, which is referred to as sulfonylurea receptor. SUR is sulfonylurea receptor. So the glucose levels are increased, taken up by the beta cells through GLUT2 sensing mechanism. Glucose inside is oxidized, more ATP is produced. The ATP will stimulate the ABC protein, which is a sulfonylurea receptor. Simultaneously, the potassium channels are closed and calcium channels are opened. That means there is influx of calcium. This influx of calcium will cause insulin secretion, and the insulin obviously will lead to decrease in the blood glucose. Thus, there is a auto regulation of the blood glucose level. So glucose is the major stimulant leading to ATP production, potassium channel closure, calcium influx, and insulin release for the auto-regulation of the blood glucose. The major stimulant is the glucose that increases that is the first factor that is increasing the insulin secretion. So other than glucose, what other factors do we have? Let me clear the slide. So the other factors are they can be categorized like hormones, nutrients, nervous system stimulation, certain drugs, and incretin hormones. Among the hormones and nutrients, we have secretin, pancreazymin, gastrin. What do these three things do? These three levels will be increased after the food intake, which directly stimulate the insulin secretion. Then proteins and amino acids such as leucine, arginine, they also stimulate the insulin secretion. Then glucagon and growth hormone, they also stimulate the insulin secretion. So these are the hormones and nutrients that stimulate the insulin secretion. Among the nervous system, if the parasympathetic nervous system and the beta adrenergic stimulation will cause increase the insulin secretion. Drugs like tolbutamide will stimulate the insulin secretion. Hormones called as incretins, they also increase the secretion. So the first one is glucose, then secretin, pancreazymin, and gastrin, proteins, amino acids like leucine, arginine, and nervous system stimulation, drugs like tolbutamide, incretin hormones. But the question arises about the seventh one, that is the glucagon and the growth hormone. How do glucagon and growth hormone act as the factors increasing the insulin secretion? Is that first coming to the glucagon? Glucagon raises the blood glucose by increasing glycogenolysis and gluconeogenesis. We have seen this in the part 1. This will result in increase in blood glucose. So what happens if we increase the blood glucose? Blood glucose will get inside, get oxidized, ABC protein activated, potassium channels closed, calcium channels opened, and insulin is released. Thus, indirectly, it increases the insulin secretion. That is how glucagon will increase the insulin secretion. Then what about growth hormone? Growth hormone is a diabetogenic hormone. It decreases glucose uptake in muscles and fat. It increases lipolysis. That means increased free fatty acids. And then there is increased hepatic glucose output. All these three factors will cause insulin resistance. So to compensate this insulin resistance, the pancreas will secrete more insulin. So this is how both the glucagon and growth hormone will be increasing in the insulin secretion. So next, going to the factors that are decreasing the insulin secretion. Under stressful conditions, or exercise conditions, adrenal medulla will release the epinephrine. This epinephrine will directly inhibit the insulin release. And the additional function is that it mobilizes the glucose from liver. We have already seen epinephrine acts by increasing the glycogenolysis in the part 1. So glycogenolysis is the breakdown of the glycogen into glucose, and the glucose from liver is mobilized by this epinephrine. So that is one factor that decreases the insulin secretion. Then somatostatin. Somatostatin is released by the delta cells of the pancreas. It inhibits all insulin action, glucagon action, and the growth hormone production or secretion. Since glucagon and growth hormone are indirectly favoring the increase in the insulin secretion, even glucagon and growth hormone are also inhibited by the somatostatin. And finally, alpha adrenergic stimulation. Alpha-adrenergic stimulation will also reduce the insulin secretion. Beta-adrenergic will increase, alpha will be decreasing. So next, this is a degradation. We have already seen the half-life is only 5 minutes. In the liver, there is an enzyme called as insulinase, which degrades the insulin. Then now coming to the most important section, that is the mechanism of action of insulin. So this is the cell membrane, as you can see, the phospholipid, biphospholipid layers, and this is the insulin receptor. This whole structure which I am marking in the red is the insulin receptor. These insulin receptors have four subunits, two alpha subunits and two beta subunits. So two alpha subunits and two beta subunits. The two alpha subunits are situated extracellularly outside the cell, and the two beta subunits are in the membrane. They span in the membrane, cell membrane, and also extend into the cytoplasm. Throughout the membrane, it is present and extending into the cytoplasm. So now you know there are receptors, insulin receptors, two alpha receptors, two alpha subunits and two beta subunits, four subunits for the insulin receptors. So the insulin comes and binds to the insulin receptor, which is a glycoprotein at the alpha subunit. The internal portion of the beta subunit possesses tyrosine kinase activity, which acts as a on switch for triggering the internal cell signals. So there the cellular cascades happen. Once the insulin binds to the receptor, it sets off multiple protein activation cascades. Many protein activation cascades happen. The primary objective is to facilitate the entry and storage of energy through the following actions like GLUT4 translocation. GLUT4 translocation, it comes to the cell membrane and it allows the sugar, the glucose, to flow inside the cell, and then it promotes the breakdown of the glucose, that is glycolysis is increased, synthesis of storage molecules like glycogen and fatty acids are also increased. The ultimate result of this signaling is a significant increase in the glucose uptake by the target tissues. So insulin is binding to insulin receptor, tyrosine kinase activity is activated, a series of protein cascade reactions will happen that will cause the GLUT4 transporters translocated to the membrane. These GLUT4 transporters will help the glucose to enter into the cell, where the glucose is converted into glycogen. Glucose is broken down, fatty acid synthesis also happens. And now coming to the clinical link, the obesity and type 2 diabetes. The effectiveness of this entire system depends heavily on the number and the sensitivity of the receptors. How many number of insulin receptors are present and how well they are receiving, how well they are sensitive to the insulin. First, let's see about the obesity. In cases of obesity, the total number of receptors will decrease. So in obesity, receptors number will decrease. So insulin will not be able to act properly. Sufficient uptake will not be there. Sufficient glucose uptake is not there. So extracellularly in the vessels itself, the glucose will remain. Then coming to the insulin resistance. This reduction causes the target tissues to become less responsive to the hormone. So obesity, the receptors number is decreased, and insulin resistance, the receptors sensitivity is decreased. So in type 2 diabetes mellitus, when the insulin signaling fails, either through fewer receptors or weakened internal messages like decreased sensitivity, it results, that is the insulin insensitivity, which is the hallmark of the type 2 diabetes mellitus. So this is the mechanism of action of insulin through the insulin receptors. What will happen if the receptors number is decreased, and what will happen if the sensitivity is decreased? Ultimately leads to type 2 diabetes mellitus. Then coming to the signal transduction. The moment the insulin binds to its receptor, it initiates a molecular relay that tells the cell exactly how to behave. This signal transduction process occurs in a few distinct phases. First, the process begins when the insulin attaches to the alpha subunits. This connection triggers the tyrosine kinase activity within the beta subunits. As a result of this, the beta subunits undergo autophosphorylation, switching the internal signal, switching on. We said the tyrosine kinase activity acts as a on switch. So autophosphorylation will lead to the activation of the uh, signal transduction. Once the receptor is active, it phosphorylates the molecules known as insulin receptor substrates. Insulin receptor substrates. Insulin has bound to the receptor on the alpha subunit. Because of this binding, tyrosine kinase activity is increased at the beta subunit, which results in autophosphorylation. This autophosphorylation will phosphorylate the molecules called as insulin receptor substrate, or the shortcut it is called as IRS, insulin receptor substrate. The chemical messenger is then transmitted to a series of serine or threonine kinases, specifically IRS1 and IRS2, which ultimately drive the cell's responses. At the IRS level, that is insulin receptor substrate level, this signal splits into two primary tracks to manage different cellular functions. The IRS1 pathway is related to growth and genes. This plays a key role in mitogenic pathways, directing the cell growth and gene expression. The growth of the cell and the expression of the genes is carried out by the mitogenic pathways, which in turn is activated by the IRS1 pathway. Then the IRS2 pathway, this branch primarily regulates the metabolic pathways. So far, we have been discussing about this glycolysis, glycogenolysis, gluconeogenesis, all these metabolic pathways are carried out by IRS2. IRS2, that is insulin receptor substrate 2. So how is the signal transducted? Insulin binds to the insulin receptor alpha subunit, beta subunit, tyrosine kinase activity is activated, autophosphorylation happens. This will phosphorylate the IRS. IRS acts via IRS1 serine threonine kinases. IRS1 will help in cell growth, gene expression, and the IRS2 will take part in the metabolic pathways. This is how insulin transduction is happening. Then next coming to the how the enzymes are being activated, like mechanism of action. Insulin operates at the transcription level, regulating the synthesis of over 100 different proteins. 100 more than 100 different proteins are activated, they're regulated, their synthesis is regulated by insulin. It essentially tells the cell which tools to build and which enzymes, which proteins to put away. The insulin increases the production of the enzymes that help process the glucose and build fats, such as glucokinase, pyruvate kinase, phosphofructokinase, and acetyl-CoA carboxylase. It suppresses the enzymes that would create new glucose, like glucose 6-phosphatase, phosphoenol pyruvate carboxykinase, fructose 1,6 bisphosphatase. These are the enzymes that are involved in glycolysis, glycogenolysis, gluconeogenesis, and all the other pathways related to the glucose metabolism. When we'll be discussing about these cycles, you will know about each of these enzymes in which reaction they are participating. Then next coming to the enzyme activation. Beyond building new enzymes, this is new enzymes synthesis or inhibition. Suppose the synthesis is happening, they should be activated. So insulin can instantly change the behavior of those already present by adding or removing the phosphate groups, phosphorylation or dephosphorylation. Through this mechanism, phosphorylation or dephosphorylation, more than 50 enzymes are regulated by this mechanism. Insulin activates protein phosphatase 1. This specific protein acts like a pair of molecular pliers, removing the phosphate groups from the inactive enzymes. Inactive enzyme. The enzymes are inactive. Some of the enzymes are inactive because there is a phosphate group, because they are in the phosphorylated state. This protein phosphatase 1 will remove this phosphate group, and because they are dephosphorylated, these enzymes will be activated. That is one thing. That is how the enzymes are activated and deactivated. Whether to remain these enzymes as phosphorylated, if they remain phosphorylated, they are inactive. If the phosphorylated, the phosphate group is removed and they become dephosphorylated, the enzyme will become active. That is the activation of enzymes. Then next, coming to the DNA synthesis and the cell growth. Insulin is also a primary driver of physical growth and cellular energy storage, anabolic hormone. So by utilizing the IRS1 pathway, insulin stimulates the DNA synthesis and promotes the cell growth. These complex pathways rely on two main messengers, that is the calcium ions and the cyclic AMP molecules. Insulin usually lowers these two molecule levels by increasing the phosphodiesterase enzymes. This drop in cyclic AMP effectively shuts down the enzymes that depend on it, like glycogen phosphorylase, preventing the breakdown of the storage energy. So insulin lowers the blood sugar not just by opening the transporters, but simultaneously ordering the cell to build more enzymes that either burn the glucose or to synthesize more glucose. This is how the insulin activates the enzymes. Then finally, coming to the physiological actions of insulin, that is the metabolic effects. Insulin acts as the body's ultimate metabolic manager, coordinating how we use and store energy from carbohydrates, lipids, and proteins. Because it primarily works to lower the blood sugar and build the tissue, it is classified as a powerful anabolic hormone. So first, let's see about the carbohydrate metabolism. In the carbohydrate metabolism, obviously, it will decrease the blood sugar. It increases the way the cell uses the sugar while simultaneously shutting down the body's internal sugar production. It increases the glucose uh, usage and decreases the production. So in the muscles and the adipose tissue, the insulin acts as a key that unlocks the GLUT4 transporter, allowing the glucose to enter into the cells via facilitated diffusion. So facilitated diffusion. This is where you have to revise the transport systems, types of transports. So facilitated diffusion, GLUT4 enters the glucose into the cells. In the case of diabetes, these GLUT4 transporters are usually reduced. But glucose entry into the liver glucose entry into the liver through the GLUT2 transporter does not require insulin. Into the cells of muscles and adipose tissue, GLUT4 transporters need insulin, whereas into the liver cell via the GLUT2, insulin is not needed. Then what happens? It stimulates the glycolysis. Insulin increases the amount and activity of the three enzymes, glucokinase, phosphofructokinase, and pyruvate kinase, to break down the glucose for energy. Then it activates the glycogen synthase enzyme to produce more glycogen. Then to keep the blood sugar levels low, insulin halts two major processes. Gluconeogenesis is inhibited and glycogenolysis is also inhibited. The enzymes that are suppressed are PEPCK, pyruvate carboxylase, glucose 6-phosphatase. If these three are inhibited, gluconeogenesis is inhibited. If glycogen phosphorylase and glucose 6-phosphatase are inhibited, glycogenolysis is also inhibited. These two pathways usually increase the glucose. So here the usage of the glucose is increased via the GLUT4, GLUT2, and production is decreased by decreasing the gluconeogenesis and glycogenolysis. So this is the effect on the carbohydrate metabolism. Next, coming to the lipid metabolism. Lipid metabolism basically it will build and protect the fat. That's how we can describe. Insulin is highly efficient at promoting the fat storage, that is lipogenesis, and lipogenesis is increased and prevent lipolysis. Fat breakdown is prevented. Lipogenesis, the insulin provides the necessary building blocks for the fat by increasing the acetyl-CoA and the NADPH and activates the acetyl-CoA carboxylase through the glycerol that is needed for the triacylglycerol synthesis or the triglyceride synthesis, which is the storage form of the fat. Indirectly, the insulin will be promoting the lipogenesis. Then it prevents the we are discussing about it like lipogenesis, now about lipolysis. It prevents the breakdown of the fat in the adipose tissue by inhibiting the hormone sensitive lipase. Hormone sensitive lipase, HSL, is also the lipase that is affected by the hormone sensitive lipase. Lipase is an enzyme. That enzyme is sensitive to the hormone. Which hormone we are talking about? Insulin. So HSL is inhibited by the insulin. In diabetes, the loss of this inhibition leads to an unhealthy spike of free fatty acids in the blood. Then the next effect is on the lipid metabolism is anti-ketogenic effect. By suppressing the HMG-CoA synthase enzyme and ensuring acetyl-CoA is burned in the citric acid cycle, insulin significantly reduces the production of ketone bodies. So lipid metabolism, it increases lipogenesis, decreases lipolysis, and decreases the ketone bodies. Ketone bodies levels also decreases. On carbohydrate metabolism, glucose level is decreased. Now coming to the protein synthesis and the cellular growth. Insulin is vital for the physical structure and the replication of the body. It promotes the synthesis of new proteins. Protein synthesis is promoted and slowing down their degradation. Insulin is required for the replication of these cells and is considered a fundamental growth factor for all the mammalian cells. So on the protein metabolism, on the lipid metabolism, on the carbohydrate metabolism, these are the physiological actions of the insulin. Next, coming to the incretins. The incretin hormones, they act as the body's digestive advance team, preparing the pancreas to handle a glucose load even before it fully hits the bloodstream. Incretins are the hormones released from the gut that significantly boosts the insulin secretion. The factors increasing the insulin secretion we have already seen, incretins. So it increases insulin secretion. When you eat, the presence of glucose in the small intestine triggers their release. They travel through the circulatory system, reach the pancreatic beta cells, prompting them to release more insulin than they would in response to the blood glucose levels alone. So, in this incretin system, there are two primary hormones responsible. There is the GLP-1 is one hormone and the other one is the GIP. So let's see about the GLP-1. GLP-1 is secreted from the L cells of the ileum. It boosts the insulin, suppresses the glucagon secretion, increases the insulin secretion, and manages the appetite. Under the healthy conditions, GLP-1 does far more than just assist with the insulin. It coordinates multifaceted approach to lower the blood sugar. That is the first thing we already seen. There is the glucagon suppression. It delays the gastric emptying, ensuring that the nutrients are absorbed more gradually. There is increased satiety, appetite control, and all these things, and therefore reduces the food intake. which ultimately leads to the lowering of blood glucose. That is GLP-1, which are secreted from the L cells of the ileum. The other one is the GIP, glucose-dependent insulinotropic polypeptide. So, glucose-dependent insulinotropic polypeptide. It is produced from the K cells of the duodenum. It stimulates the insulin release in response to food intake. In individuals with type 2 diabetes, this efficient system is compromised, typically characterized by reduced cellular response. Then the next one is the DPP-4. Naturally occurring GLP-1 is extremely short-lived because it is rapidly broken down by an enzyme called dipeptidyl peptidase 4. So DPP-4 is dipeptidyl peptidase 4. The DPP-4 is the enzyme that breaks down the GLP-1 to maintain the higher levels of active incretins. The clinicians generally use DPP-4 inhibitors such as sitagliptin and saxagliptin. So DPP-4 inhibitors are sitagliptin and saxagliptin. These oral medications block the enzyme, allowing the body's own GLP-1 to circulate longer and help to stabilize the blood glucose levels. This is the incretins. There are let me clear the slide 1 second. Two important molecules related to the incretins, that is the GIP, GLP-1, secreted by K cells in the duodenum, L cells in the ileum. The GLP-1 suppresses the glucagon secretion, slows the gastric emptying, increases the satiety, reduces the food intake, and finally lowers the blood glucose, along with increasing the insulin secretion. Whereas GIP, it reaches the pancreas, stimulates the insulin secretion, and by that it decreases the blood glucose. And the GLP-1 is degraded very soon because of the enzyme dipeptidyl peptidase 4. So the clinicians will be using DPP-4 inhibitors like sitagliptin and saxagliptin, which will increase the natural GLP-1 levels, lowering the blood glucose. Coming to the glucagon. Glucagon is produced by the pancreas alpha cells. This ensures your blood sugar levels don't dip too low. Glucagon is a polypeptide hormone consisting of 29 amino acids. Active insulin is 51 amino acids. Glucagon is 29 amino acids. It is initially produced in a longer form known as precursor, pre-glucagon precursor. There it is pre-pro-insulin, pro-insulin, and then active insulin plus C-peptide. Here, pro-glucagon precursor. Pro-glucagon precursor is processed. Finally, glucagon is produced. The primary site of action is the alpha cells located within the pancreatic islet cells. There is something called as enteroglucagon. Enteroglucagon is secreted from the intestines. It is one of the hormones that is related to the glucagon immunologically. This is the gut glucagon, enteroglucagon. The general glucagon is the pancreatic glucagon. Both of these share some common immunological characteristics. Then coming to the half-life, glucagon is also having very short half-life, that is 5 minutes. Liver is the primary organ responsible for its inactivation and removal from the system. The concentration of glucose in the blood is basically because of insulin glucagon balance. Glucose is the most significant factor controlling the glucagon release. High blood glucose, glucagon is sufficient, so inhibits the glucagon secretion. The body inhibits the glucagon secretion. Whenever there is low blood glucose, glucagon levels need to rise and the body will stimulate the glucagon secretion. Next, coming to the mechanism of action of glucagon. Just like how insulin binds to the insulin receptor, the glucagon also binds to the glucagon receptor on the plasma membrane, which will activate a protein called as G protein. The inactive G protein has GDP plus alpha plus beta plus gamma subunits, three subunits alpha, beta, gamma bound to GDP. So all these four are together. Once activated, first the GDP will be out, then alpha plus beta plus gamma, then alpha will be out plus beta gamma. After the alpha being separated, the alpha binds to GTP to form alpha GTP, which is represented here. This alpha GTP will produce an enzyme called as adenylate cyclase. This adenylate cyclase is cumulated basically. This adenylate cyclase will be converting ATP into cyclic AMP, which acts as a second messenger. So what is the function of cyclic AMP is that it phosphorylates the certain enzymes like the glycogen phosphorylase, which breaks down the glycogen into glucose. When it is dephosphorylated, it is inactive. On phosphorylation, it gets activated. Glycogen breaks down into glucose. So this is how glucagon will increase the glucose levels, glycogenolysis. At the same time, the glycogen synthase enzyme, which is active in the dephosphorylated state, when it is phosphorylated, it becomes inactive. That means glucose to glycogen is not happening. So this way simultaneously the breakdown of glycogen is increased, increasing the glucose levels, and the storage of glycogen is decreased, thereby the glucose levels remain elevated itself. So this is the mechanism of action of glucose. Coming to the physiological actions of glucagon, it is a hyperglycemic hormone, anti-insulin hormone, and the net effect is decided by the insulin-glucagon ratio. The primary target of the glucagon is the liver, where it promotes the glycogenolysis by activating the enzyme glycogen phosphorylase. That is first function, glycogenolysis is increased, and the second one is it promotes the gluconeogenesis by activating the pepsi-K enzyme, glucose 6-phosphatase, and the fructose 1,6 bisphosphatase enzymes. So this is how on carbohydrate metabolism, that is, it increases glucose, whereas insulin decreases glucose. Coming to the adipose tissue, insulin, it actually favors lipogenesis. Insulin lipogenesis increases, lipolysis decreases, ketone bodies decreases. Remember this. Why the glucagon is anti-insulinic hormone is that the glucagon will increase the breakdown. It increases the lipolysis. So free fatty acids are increased. The free fatty acids undergo beta oxidation, and therefore there will be increased acetyl-CoA, there will be increased plasma-free fatty acids, and it also increases the ketone bodies, exactly opposite to the insulin. So glucagon basically it increases the blood glucose, increases the free fatty acids, and increases the ketone bodies. So these are the actions of glucagon. So with this, we will be completing about the part 2, where we have discussed about insulin, its mechanism of action, structure, physiological functions, signal transduction, production or synthesis. Then we have seen glucagon, production, mechanism of action, and the physiological functions. In between, we have seen about incretins also, the functions. So this is the mechanism of action of the hormones. The last part of the part 1 that we have discussed has been completed in the part 2. but there is still much more to discuss about the blood glucose regulation. What happens if there is unregulated, that is diabetes mellitus? We have to talk more about the insulin resistance, the clinical features, the complications, how do you diagnose, what are the lab investigations, and how do you manage? All these things need lots of time, so we have to go for a part 3. Then we have to discuss about metabolic syndrome, obesity, and finally hypoglycemia. So kindly stay tuned for the part 3. It will be uploaded very soon. Thank you for your patient listening.